role in disease or in physiological situation. Okay, so microRNA, what is the length? Around 20, 200. Okay, और microRNA का लेंथ क्या है? नहीं पता. Okay, micro means small. कितना स्मॉल हो सकता है कोई आरएनए टू बी फंक्शनल? 200 काफी बड़ा है. माइक्रो नहीं बोल सकते, 200 स्मॉल बोल सकते हैं. 3 बेसेस, 3 बेस वो फिर एक कोडन है. बट माइक्रो आरएनए आर मोस्टली 19 to 22 बेसेस. ओके, उससे कम होगा तो फिर उसका मीनिंग ही खत्म हो जाएगा इसके लिए. ठीक है, बट दिस माइक्रो आरएनए जो मैच्योर माइक्रो आरएनए होता है, that is like 19 to 20. But when the microRNA is actually transcribed from the genome, it is like 70 bases around. So it is like a loop structure. Okay, and by processing, it is processed into a smaller fragment, either from this arm. If it is from the 5' end, we call that 5p. If it is on the 3' end, we call that 3p. So if you see the microRNA database, you will see, suppose microRNA 1, okay, you will see hyphen 5p or 3p. So that denotes from which side the microRNA is coming. The parent microRNA is 1, but it can give rise to 2 microRNAs, and one of them is usually more abundant and functional, while the other one is expressed in little bit amount. Okay, so sometimes 3p is more abundant, sometimes 5p is more abundant. So just saying microRNA number doesn't mean much unless you specify 5p or 3p. यहाँ पे जैसे लिखा है, it has, if it is coming from this side, it has to be 5p, from this side it is 3p, and both have complementarity. So the function could be completely different because the sequence are completely different from each other. So the target genes will be different. And when it is transcribed as a whole, then different fragments are chopped off by different enzymes during the processing to give rise to the final microRNA. Okay. So who discovered microRNA? नहीं पता. और कब डिस्कवर हुआ ये किताब में है. माइक्रोआरएनए है ना? हाँ, ये सही बोल रहे है. हाँ, तो फिर बताओ कब डिस्कवर हुआ था? नहीं पता. ओके, 93. Okay, so it was discovered in C. elegans. Okay, and it was lin 4 microRNA, and that went for the Nobel Prize, right? And microRNA binding sites are present in 30% of the genes. They are like present in all genes, but 30% of the genes are actually regulated by microRNA. They are small, but they regulate huge number of genes at a given time because of their complementarity. Okay. So microRNA transcribed from the gene through transcription factor, they can regulate the target gene, or this transcription factor can directly regulate this. And in the other way, microRNA can inhibit the target gene, or it can inhibit the transcription factor, which actually like promote this one. So it can have an opposite effect. Okay. In the other way, microRNA can inhibit or promote transcription factor which regulate target gene. So all possible ways microRNA can regulate the gene expression directly through the gene by interacting with the gene, or through the transcription factor. Okay. So when we say microRNA biogenesis, as I said, it is a big loop getting transcribed from the genome, then getting processed. Okay. So the first step is transcription from the DNA with the transcription factor and pol 2. Okay. So how many RNA poles are there? 3, right? 5. 1 2 3. ओके, तो 1 क्या ट्रांसक्राइब करता है? 2. 2 is mostly for the mRNA and microRNA, link RNA also, and 2 is here transcribing the whole length of the microRNA gene. That then folds because of the complementary sequence. It makes the hairpin loop, and then that hairpin loop is processed by DGCR8 and DROSA. So it chops up the ends here. So you have only the stem loop without the tails. Okay. Now your exportin 5 comes into play. So it's a smaller version now, and this exportin 5 exports this small hairpin loop to outside the nucleus. In the nucleus, into the cytoplasm from the nucleus. So in the cytoplasm, your dicer comes into play. So you have a duplex stem loop now, and dicer process that into a duplex but without the loop. Now you have 2 strands, one is your 5p, one is 3p. Based on the target, the 5p or 3p can be loaded into the RISC complex. RISC का full form क्या है? Anyone? नहीं पता? siRNA mediated RNA induced silencing complex. Okay, so that is required, that complex is required for microRNA mediated regulation as well as for the siRNA mediated regulation of the genes. So based on the 5p or the 3p, they have seed sequences. Seed sequences are at the 5 prime end of the microRNA, which can target the target mRNA, and mostly in the 3 prime UTR, and with the RISC complex the seed sequence binds. But many times even without a perfect seed sequence, if it has multiple sequence throughout, like it is a 20 base sequence, the seed sequence is like 2 to 8, so like 7 bases it binds. But sometimes like you have 5 bases at the seed sequence, but the other end 3 prime end, that also have another 5 bases binding. So you can have like half binding this side, half binding this side, you have a loop in the middle. So the way microRNA is shown, like sometimes binding half here, then a loop, then binds here. So that also works. Okay, so like it should bind like 7 8 base should bind to the mRNA, so that it can act on the mRNA through the RISC complex. So but there are microRNAs who doesn't follow the regular pathway. Like the regular means what I said, like transcribe, then process through BROSA, and then go through dicer. But some microRNAs, they do not follow the exact pattern what I just said. They skip something like DROSA bypass, and some they go for direct cleavage by AGO2. AGO2 is actually involved in the RISC complex for the silencing, but sometimes that also work for the microRNA processing. So that is non-canonical pathway. And when it comes to the function, microRNA binds to the 3' UTR, then degrades the nascent protein. It induces degradation of the poly-A tail. It can inhibit the cap, okay, so that the translation doesn't happen efficiently, or it can inhibit the or slow the elongation. So in the last class also I said all this. So by all means, microRNA can induce degradation of the mRNA, not through direct cleavage, but by inducing the poly-A tail degradation, so that the mRNA is not stable. It is not a like degradation directly by the microRNA, but indirectly by depleting the poly-A tail, and then protein by inhibiting the translation. All that happens when the microRNA binds to the 3' UTR, right? But do you think microRNA binding sites are not there in other places? Because it is always shown in 3' UTR, all images, right? I have been showing it is working in the 3' UTR. Actually, microRNA binding sites are everywhere, okay? But only the 3' UTR one is more effective, and others don't work that efficiently. You know why? ऐसा क्यों होगा?
Because of the presence of poly-A tail.
Because no, microRNA is at the 3' UTR, poly-A tail is downstream to that. But you have microRNA binding sites in the 5' UTR in the coding region also. But they don't function that well compared to 3' UTR, right? The question is why? Even if you have binding sites in the coding region, they do not efficiently like stop the translation or inhibit. ऐसा क्यों होगा? मैं नहीं बताऊंगा, जल्दी क्लास खत्म करना है, हाँ? बताओ। हाँ क्यों? हाइपर हाँ, CDS में नहीं होगा, बाइंड हो सकता है, but your ribosome is moving, right? So it may not allow the complex to build on there. But 3' UTR is relatively free. Ribosomes are not moving. So RBPs or rich complex, all that can bind there because your ribosome falls off after the stop codon. So the 3' UTR can act as a regulatory part. Okay. So then microRNA binds and regulates the gene expression. And then how does microRNA itself is regulated? It has to be degraded ना, कैसे degrade होगा? कोई विकेट नहीं, कहाँ चला गया? What happened? Okay. Ubiquitination, that is one way, and the other way is degradation by the exonuclease. So every RNA, they either endonuclease or exonuclease, they chop them up. So depending on their stability, they are degraded. And then how do you detect them in a system? So if the microRNA are there, like we said, for the regular RNA, you do PCR, or for circular RNA, you put primers facing outwards. For microRNA, these are very small, 20 nucleotides. So primers are usually 20 nucleotides. So you cannot put two primers to amplify, right? How will you do that? स्लाइड देख के मत बताओ कैसे करोगे? टोटल 19 न्यूक्लियोटाइड है, अभी पीसीआर करना है, कैसे करेंगे? ऐड ए पॉलीएटेल, mRNA में पॉलीएटेल होता है, सो जस्ट ऐड ए पॉलीएटेल एंड देन फॉर द cDNA synthesis use oligo-DT with a universal sequence or adapter sequence that will act as your reverse primer. Then forward primer is your microRNA primer. You can amplify. The other way is ligate adapter to the end of the microRNA, so that you have the microRNA and the adapter sequence. So now you can make the cDNA with the adapter sequence, any oligo complementary to your adapter sequence, and then make the cDNA, then go for the PCR. Or the other option is have a stem loop primer which can be longer and a small stretch of the oligo binding to the 3' end of the microRNA, and that can extend through the cDNA synthesis. So you have a longer product now, cDNA, that can go for the PCR amplification. Okay, so multiple ways are there, and the other like important way to discover microRNA is by sequencing, like we discussed. Like you add the adapters. For this one you don't need to fragment it again, it is already small, so just add the adapters, amplify them, go for sequencing, and then match with the genome, and you find the microRNAs. So what we discussed is basically how microRNA is transcribed, processed in the nucleus, then in the cytoplasm through dicer processing, it loads into the RISC complex and then act on the target mRNA. Okay, mostly they are inhibitory, but there are instances where microRNA can upregulate translation also. It is not always inhibitory. Okay, sometimes like I have one publication where we saw the microRNA binding site in the 5' UTR, not in the 3' UTR, 5' UTR, and it was found to upregulate the translation of the target. Okay, so it is not always like microRNA will inhibit. Okay. And one of the system my lab works on is muscle. You know muscle, right? So how muscles are formed? Any idea? No. Skeletal system. You have satellite cells in the muscle. Those are stem cells. They get activated once you exercise or there is an injury. Okay. In both cases, your stem cells get activated. They give rise to more number of muscle cells. They proliferate. They fuse together to form myotube. But with aging, muscle mass and the activity, strength and mass goes down, right? And that happens because this process, the activation of the stem cells and the renewal or the formation of muscle fiber, they all have gone down because of the aging process. So genes are not expressed well, so the whole process is slowed down, so you have less muscle. So with age you increase your strength, muscle mass, and then they slowly go down. And as you can see here, there are very many transcription factors which are involved in this whole process. So from the satellite cell to myoblast, then elongated myocytes, and these myocytes then they fuse together to have multinucleated myotubes, and many myotubes they form the myofibers, and those form the muscle in this body, and they are activated with exercise or injury. And there are I said transcription factors, and some of these transcription factors have been reported to be regulated by microRNAs. So if the microRNAs are upregulated or downregulated, they can change the expression pattern of the transcription factors which are involved in myogenesis. That's how they can regulate the myogenesis process. Okay. So as I said, in the young and old, so this is a old work from my postdoc lab where we looked at young and old muscle cells, and the previous publication in the lab they showed, okay, one of the microRNA clusters, these are two microRNAs coming from the one microRNA, primary microRNA, one gene giving rise to two microRNA, they are upregulated. So we wanted to see if something is upregulated in the old muscles, why it is up and how it is affecting the myogenesis process. So we took a cell line, this is C2C12 myoblast, you differentiate them into 6 days, you will see myofibers. So basically now here you see single cells, they grow, then when you induce them to differentiate in the culture plate, you see muscle fibers, long multinucleated fibers by 6 days. So that is myotubes basically. So in these cells, what we did, we inhibited, transfected the microRNA. Two microRNAs were there with us, and one of them you see very little myotubes where you can see myotube here and in the control. So this suggests that this microRNA 451 could be an inhibitor of myogenesis because you don't see myotubes there, and this is happening, and this is another way to show the creatine kinase activity. So this is an enzyme that is expressed during the myogenesis process. So in case of 451, you see a decrease in the activity. So that shows myotube formation is inhibited with that microRNA. Then how do you find the gene? Okay, so if the microRNA is functional, the other microRNA was not functional. Like in the morning I said, some things are expressed but they may not be functional actually. So the other microRNA didn't do anything, but this one inhibited myogenesis. So we wanted to see what are the genes they are regulated through this microRNA. We predicted the genes targeted by this microRNA and we found a few of them, and one of them is this SPARC. Okay, that's a protein involved in myogenesis, and when we overexpress this microRNA, okay, the SPARC mRNA goes down. So basically this is inhibiting the mRNA expression, and as I said, like it could be two ways. One is the stability, the other one is the translation. So to see whether this microRNA is affecting the translation of SPARC, we did polysome fractionation. The way I said in the morning, you have sucrose gradient where the heavy polysomes settle at the bottom, the lower ones go up, and you fraction them and you see low molecular weight polysomes, high molecular weight polysomes towards the end, and this is how you see, and each peak in the polysome fractionation actually represent one ribosome. So if you have one this is one 2 ribosome, this is 3, this is 4, this is 5, this is 6, like that. So number of ribosomes increase on the mRNA, they will be stacked in a specific position on the sucrose gradient. So the more towards the end, you can assume they are more translating. Okay, so here we have the microRNA and you see a inhibition, like you see the peak here in the control. With the microRNA there is a decrease in the peak towards the end and increase in the peak at the beginning, like on the top. So that shows that the microRNA is actually inhibiting the translation of this protein. And then the other way to check the translation efficiency is luciferase assay. Have you heard luciferase assay? Right. So it is a reporter assay. So you have a reporter gene, you have 5' UTR, 3' UTR, then you put your microRNA of your choice and see how much luciferase is getting expressed compared to the control. And that way you don't need an antibody for the gene. What you need is the luciferase reporter and the reagents to see the light. So you transfect the construct, you put the microRNA with or without the control or the inhibitor, and then look at how much light it is emitting in the lysate after the transfection, and like experiment is done for like 24 hour or 48 hour, you check the how much luciferase activity is there. So that way we also confirmed this microRNA targets the luciferase gene and inhibits. So that way you confirm the microRNA is actually targeting the 3' UTR and inhibiting its function. And then in the differentiated cell, like the single cell, the myoblast and the myotubes, those are differentiated cells. So spark mRNA goes up, and here is the western. Then when you put the microRNA here, that inhibits the spark expression. So this way we conclude that like when you move from myoblast to myotube, this microRNA level goes down, that induce spark expression. So, and that is required for myogenesis. So this is one of the example from the lab, like we published long time back. So this is one way microRNA can regulate myogenesis. And as I already said, like microRNA doesn't always inhibit. This is another old publication from my data. So microRNA can also increase the expression, and this was in the 5' UTR of insulin gene itself. So that microRNA increased the expression. Okay. So it's not always microRNA, if it is there, it will inhibit. So depending on where it is binding and the mechanism, things could be different. Okay. And all this happens because of RNA-RNA interaction, right? So we are talking about microRNA, but there are other RNAs. These are small nuclear RNA, snRNA, that is important for splicing, right? Then tRNA, important for translation. MicroRNA, translation, stability, link RNA, translation, stability, splicing, and then circular RNA, mRNA, microRNA interaction is for the protein mRNA stability and translation. Now, recently this is from our lab, like 2023, we published a review on microRNA regulation by circular RNAs. Okay, so what happens is anytime you have microRNA and the mRNA, they suppose you assume microRNA is inhibiting, so you have low microRNA here, you have high number of mRNAs, or in the other condition you have high microRNA but you have high number of circRNAs. So they all are sponged, they are expressed but they don't work. Okay. In the opposite scenario, you have excess of microRNA that can inhibit expression, or you have less number of circRNA, so the microRNA is more active. So in all two different completely opposing scenarios, you can control gene expression depending on microRNA availability and circRNA availability, because circRNA is acting as a sponge for the microRNA. So if you are saying microRNA goes up, that doesn't always mean your target will go down. Okay. If another circular RNA which is binding to your microRNA, that is upregulated. So microRNA will not have any effect, right? It is your microRNA is going up, but circRNA is also going up to stop its activity. Okay. So we'll see one more example in muscle cells again. Okay. So in aging scenario also, we wanted to see, we saw how microRNA is regulating myogenesis. Now we'll see how circular RNA is regulating myogenesis through regulating microRNA. So what we did here is we could not take young and old human muscle, but we took mouse muscle, young and old mouse muscle, then did the sequencing the way I explained in the morning. You remember, we did NGS, found circular RNAs, and then look at their expression, whether they're going up or down in young and aged mice. You find some are exclusively in the young, some are in the aged mice, and then look at how big the circRNAs are. Majority are below 1000 bases. Okay, and some are very long circRNAs, but majority are within 1000 bases. And then how do you validate them? By divergent primers, right? So you put two primers facing outwards on the linear RNA, but they face inwards when you talk about the circRNA. And then you do the PCR, get the PCR product, and then do Sanger sequencing to validate if you are getting the junction sequence like the last one I described. And then to validate whether the circRNA are genuinely circular or they are coming just because of the splicing error, what we do is do a treatment called RNA-CHAR. So this is an exonuclease that is mostly used to validate RNA is a circle or it's a linear one. So it will degrade from 3' end to 5' end. So the linear RNA will be degraded, but the circular RNA will remain the same, right? So they will not be digested. As you can see here, GAPDH mRNA, it is degraded. Okay, it's in a logarithmic scale, 1 is here. So this is below 0.1. Okay, but your circRNA are not changing much. So because you are not degrading them. So this is one way to confirm that whatever PCR amplicon you are getting, they are genuinely coming from the circles, not from the linear RNA. Okay. And in the aging muscle, what we did is like find out, validate the different markers of aging, P16, P53, and then look at the circRNAs we validated by PCR. So most of them didn't change. One of them changed significantly, but one of them you see here, NFIX, was very highly abundant in the cell in young to old muscle. Then we is doing like if something is very abundant in the muscle, we looked at whether this circular RNA is conserved or not. First thing, okay, if something is expressed in mice, we need to see if it is relevant for the human, right? So we looked at the human cell line. So these are primary human myoblasts differentiated into myotubes, and the color you see, this is a immunostaining. Anyone knows immunostaining? No? So basically, like if you want to see a protein expression in the live cells, you fix the cells with antibody, and then the secondary antibody should have a label. Okay, so that you read the color in the microscope, take the image and see that. So, and the nucleus is stained by DAPI, so that stains the DNA, so you get that blue color. The myotube, this is with MYSC antibody, so that is a marker for the myotube, muscle marker. So you see the myotubes formed, and then we looked at the sequence. Mostly the circRNA sequence is same. in both human and mouse. Then look at how is it regulating. It should be binding to some microRNA, right? So we took mid-dB predicted microRNAs target scan. These are the microRNA expressed in the muscle, and mid-TAR base. In the morning I mentioned mid-TAR base, right? So mid-TAR base is a database that has microRNAs which have been validated by some biochemical method, whether it is sequencing or luciferase assay, Western. So validated functional microRNAs. So from all this, we got like 15 microRNAs which could be targeted by our circRNA and could be regulating myogenesis, right? So what we need to do now? microRNA silencing और circRNA silencing, क्या करेंगे? microRNA silencing. No, our focus is the circRNA. circRNA targeting this many microRNAs, right? So we need to see which microRNA can be functional. So we looked at the literature, because you cannot inhibit all microRNA to see which one is functional. So look at the literature, which one is already known. All are known for functionality. So but which one is actually having a function in the muscle? So one we found is 204 that regulate myogenesis, and the target is MEF2C. So this is myocyte enhancer factor 2C, that is a transcription factor, which is a target of that microRNA. So this is a direct target of the microRNA. So if we silence circRNA, what will happen? MicroRNA will not upregulate. MicroRNA activity will go up. We are not regulating microRNA expression. We are regulating microRNA activity. Your circRNA is inhibited, like not expressed. So microRNA is free to act on the target. So the target is MEF2C, which is a transcription factor required for the